Epigenetic targeting in pediatric brain tumors. My name is Michael Goldstein. I'm a radiation oncologist and a basic scientist at the Johns Hopkins University. I would like to thank everybody for coming today, and I would also like to thank the organizers for inviting me to present the research from my lab here at this wonderful conference in San Diego. We're going to talk about the role of the histomark H3K27me3 in radiation response in pediatric brain tumors. First, I'm going to talk about the DNA damage response in general. Then we're going to focus on the role of H3K27 trimethylation in radiation response in a pediatric brain tumor called medulloblastoma. Then we're going to look into the molecular mechanism of radiation resistance in H3K27 trimethylation deficient medullo. And then finally, we're going to talk about the strategies that we developed to overcome radiation resistance in these tumors, and also the implications for other pediatric brain tumors. Our cells have evolved to cope with DNA damage that arises on a daily basis from many different sources. And the way the cells respond to DNA damage determines their fate, which can lead to activation of the cell cycle checkpoints, change in the transcriptional program, DNA repair, and also induction of cell death. There are many different DNA lesions, including basic sites, crosslinks, and DNA breakage, including DNA single-stranded breaks and DNA double-stranded breaks. The reason why DNA damage and DNA damage response is so fascinating for clinical oncologists like myself is that we actually use radiation and chemotherapy in order to induce DNA damage in cancer cells when we are treating tumors. And radiation and many different types of chemo induce different DNA lesions, and there are specific DNA repair mechanisms that can recognize and repair those lesions. For example, in case of radiation, the most common lesions are oxidative damage, followed by single-strand breaks and double-strand breaks, with the double-strand breaks being the most toxic lesions that are ultimately responsible for the tumor cell kill. So why do epigenetics matter in DNA damage response? Epigenetics refers to any kind of modification of the chromatin structure or its chemical composition that does not change the sequence of the DNA. And it can occur locally at the site of DNA lesions or globally, resulting in global epigenomic changes. And epigenetic changes can be different at baseline and then affect the subsequent DNA damage response, or they can be triggered by the DNA damage. Here are several mechanisms illustrating how epigenetics can be involved in DNA damage response signaling. For example, the ATM kinase that is activated in the double-strand breaks can phosphorylate histone H2X, resulting in formation of gamma H2X domains. And histones can be ubiquitinated by 3 ubiquitin ligases, such as RF168, with this process being important for recruitment of downstream DNA damage response factors. While the epigenetic changes occurring at the double-strand break site are quite well studied, the baseline epigenetic changes affecting DNA damage response are less well understood. In this context, we focused on the histomark H3K27 trimethylation and its role in radiation response in medulloblastoma. Medulloblastoma is a malignant pediatric brain tumor that arises in the posterior fossa, in the area of the cerebellum, and can metastasize to the spine. This is the overview of the historic treatment of medulloblastoma. It was first described in the 1920s. In the 1950s, radiation was added as an adjuvant treatment to surgery. Then in the 1970s, chemotherapy was first utilized. In the 2000s, we got the first molecular characterization of different subtypes of medulloblastoma. And then over the past 10, 15 years, there's been a literal explosion of molecular data further characterizing the transcriptomic and epigenetic traits of this tumor. The classic molecular subgroups of medulloblastoma are Wnt, sonic hedgehog, group 3 and group 4, that differ with regard to their biology, but also to treatment response. The Wnt tumors, for example, can be very well cured, whereas sonic hedgehog and group 4 tumors have an intermediate prognosis, and group 4 tumor patients have very poor prognosis, with over 50% of them dying from this cancer. Interestingly, more recent studies have revealed that there are even more molecular subgroups. Some of them can actually overlap between the classic group 3 and group 4 subgroups. This is the current treatment paradigm. The patients first undergo surgery, and then they're treated with radiation and chemotherapy. When we treat them with the radiation, we treat the entire craniospinal axis, including the brain and the spine, followed by a radiation boost to the tumor bed. Importantly, the molecular classification has not really affected our clinical decisions and is not really used for treatment decisions in the routine clinical practice. One of the issues with medulloblastoma is that some tumors are radiation resistant, resulting in treatment failure, tumor relapse, and these patients have very poor outcomes. And we have to say that so far, molecular profiling has failed to identify mechanisms of radiation resistance, and there are also no specific markers of radiation response in medulloblastoma patients. In this slide, we looked at the histomarker H3K27 trimethylation that was shown to vary strongly within the group 3 and group 4 tumor samples. However, nothing has been known about its role in treatment response. H3K27 trimethylation is a transcriptionally repressive histone mark that is induced by the EZH2 histone methyltransferase. And so what we did, we took group 3 and group 4 medulloblastoma patient samples, and we stained for H3K27 trimethylation with H3 serving as an internal control. And so here are samples of the trimethylation deficient and of trimethylation proficient samples. We found that roughly half of those tumors were trimethylation positive and half were trimethylation negative. We then wanted to know if the H3K27 trimethylation status correlates with treatment response and survival. And strikingly, we observed that patients who had a loss of H3K27 trimethylation mark did really poorly, resulting in lower progression-free survival as well as overall survival compared to the patients who had a high expression of the H3K27 trimethylation. Then we wanted to know what is the molecular mechanism behind this. So we took it back to the lab, and we used a medulloblastoma model that was first developed in Martine Russell's lab at St. Jude. It is a group 3 medulloblastoma model that utilizes mutant GNPs that are 53 and CDK and to C null and that overexpress MYC. And as you can see here, they can form tumors in the cerebella of mice that transcriptionally are very similar to human group 3 medulloblastomas. And importantly, those tumors can also be taken out of the mouse, and the cells can be cultivated ex vivo and be used for mechanistic analysis. By knocking out EZH2, we mimicked an H3K27 trimethylation deficient phenotype. We then exposed the cells to radiation, and we found that the H3K27 trimethylation deficient cells, shown here in red, are significantly more resistant to radiation than the H3K27 trimethylation proficient cells. We then wanted to know what the reason was for this radiation resistance. So we looked at different potential mechanisms of radiation resistance, including increased DNA repair, and we found that there was in fact no difference in double-strand break repair between those cells. We also found that the H3K27 trimethylation status does not affect the radiation-induced cell cycle arrest. And then finally, when looking at different cell death pathways, including apoptosis and autophagy, we didn't observe any differences with regard to the H3K27 trimethylation status. Finally, we looked at pro-survival pathways, including the ACT and the MAC pathway. And what we observed was that in the H3K27 trimethylation deficient medulloblastoma cells, there was an earlier and a more excessive activation of the pro-survival ACT signaling pathway after radiation exposure. There was also an activation of the MAP pathway, but here the trimethylation status did not matter. We then inhibited the ACT pathway either using direct ACT inhibition or PI3K inhibition, showing that while it didn't have an effect on the H3K27 trimethylation proficient cells with regard to the radiation response, inhibiting ACT was able to rescue and mitigate the radiation resistance in the trimethylation deficient cells. As we started looking for the mechanistic explanation of the excessive ACT signaling in the trimethylation deficient medulloblastoma cells, we found that an epigenetic switch occurs in the cells from the trimethylation to an increased H3K27 acetylation. So we hypothesized that this epigenetic switch from the transcriptionally repressive trimethylation to the transcriptionally activating acetylation could increase the expression of an ACT pathway stimulator that can then result in radiation resistance. First, we verified this hypothesis by reducing the excessive H3K27 acetylation in the trimethylation deficient cells using a P300 inhibitor, which is a histomethylase transferase responsible for H3K27 acetylation. And we showed that P300 inhibition was able to mitigate radiation resistance in the trimethylation deficient cells and rescue radiation response. Then using ChIP-seq, we looked at the epigenetic dynamics within the promoters of genes that are known to affect the ACT signaling pathway. And we specifically were looking for genes where the loss of the trimethylation is accompanied by an increase of H3K27 acetylation within the gene promoter. And we identified two such genes, EphA2 and PAK3CB. Then using ChIP, we verified the results, and in fact, we observed that drop of the trimethylation is accompanied by hyperacetylation within the EphA2 gene promoter. However, we were not able to verify these results for the PAK3CB gene. EphA2 is a tyrosine receptor kinase that has been known to stimulate ACT signaling. We show that consistent with ChIP-seq results, the transcription of the EphA2 gene was upregulated in the H3K27 trimethylation deficient medulloblastoma cells, resulting in increased EphA2 protein levels. Then using IHC, we determined EphA2 expression levels in medulloblastoma patient samples. And we showed that EphA2 was highly overexpressed in H3K27 trimethylation negative tumors, and it was not expressed in H3K27 trimethylation positive tumors, which was consistent with our results in our cell-based model, demonstrating that EphA2 is overexpressed in cells that lack H3K27 trimethylation. Next, using EphA2 inhibitors, we demonstrated that by inhibiting EphA2 activity, we can reduce the hyperactivation of the ACT signaling pathway that we observed in following radiation in H3K27 trimethylation deficient medulloblastoma cells. And then we also show that inhibition of EphA2 mitigates radiation resistance in trimethylation deficient cells. We then looked for a clinically translatable strategy to blunt the H3K27 hyperacetylation and therefore rescue radiation response in the trimethylation deficient cells. And we chose to use BRD4 inhibitors since BRD4 proteins are required for the P300 histone acetyl transferase to exhibit its activity. As you can see here, BRD4 inhibitor JQ1 can strongly suppress H3K27 and H3K57 as 56 acetylation levels. Here we show that in our model, JQ1 was able to effectively suppress H3K27 hyperacetylation in trimethylation deficient cells. It also strongly reduced EphA2 expression, and as you can see here, it reversed the excessive activation of the ACT signaling pathway in the trimethylation deficient cells following radiation exposure. And we showed this in murine medulloblastoma cells as well as in human medulloblastoma cells. And finally, we show that BET inhibition is very effective at mitigating radiation resistance in the H3K27 trimethylation medulloblastoma cells and sensitizing them to radiation. Here is a model that we developed based on our results. We show that in the H3K27 trimethylation proficient medulloblastoma cells, the expression of the EphA2 gene is silenced, which prevents excessive ACT signaling following radiation exposure so that the cells are sensitive to radiation. However, if the H3K27 trimethylation histone mark is lost, this results in a hyperacetylation of chromatin, which then upregulates the expression of the FHA2 gene, which in turn drives excessive activation of the pro-survival actin pathway, thus conferring radiation resistance to the H3K27 trimethylation deficient medulloblastoma cells. And then we show that by inhibiting the activity of the CBP300 using BAT inhibitors and therefore reducing the hyperacetylation, we can mitigate this radiation resistance. Currently, we're screening the all available BAT inhibitors to identify the most effective ones in H3K27 trimethylation deficient medulloblastoma. They can be combined with the radiation, with a plan to test them in vivo models of medulloblastoma using craniospinal radiation, and then finally go into clinical trials where we anticipate to combine BAT inhibition with radiation therapy in medulloblastoma patients. Interestingly, loss of H3K27 trimethylation is not only common in non-WNT, non-spinal-catching medulloblastoma, as we just demonstrated, but is also characteristic for other pediatric brain tumors, including PFA ependymoma that has low H3K27 trimethylation levels due to an overexpression of the protein called EZHIP, which acts as an EZH2 inhibitor. Further, diffuse midline gliomas have low H3K27 trimethylation levels due to an H3K27M mutation. These tumors are characterized by a very aggressive growth, and they are very hard to treat and are commonly resistant to radiation. However, nothing is known about the role of H3K27 trimethylation loss in PFA ependymoma and diffuse midline glioma in radiation response. Therefore, we are currently investigating the role of this histone mark in radiation resistance in PFA ependymoma and DMG. Finally, I would like to thank the members of my lab who were responsible for this work. This was mainly carried out by Nishan Gabriel and Kumaresh Balaji, and now Nita Verma is taking over this project. And I would like to thank some of the foundations that sponsored this project, including the Andrew McDonough B+ Foundation and the Collaborative Rally Foundation. And thank you very much for your attention.